All right. Hi, everyone. Welcome to our 18th ESCOM webinar in the series of the 2026 ESCOM webinars we've had so far. It's a pleasure to have you all here, both people who are here in person and people watching later on our YouTube channel. Today's webinar will specifically be about beach vegetation. It's the end of May. While the weather may not agree with it, it is warming up. And as the weather warms up, you may see as you go walking on the beaches, there are little plants here and there sprouting up and down the beach. That you may be wondering, what plants are on the beach? You may be wondering, what's the distribution of those plants like? And so today we have Dalhousie student Kaitlyn Quinn here, who's done a thorough analysis of beach vegetation on our Eastern Shore beaches. So yeah, Kaitlyn, thank you so much for being willing to present this webinar.
Thank you so much for the introduction, Everami. Just so you know, I am currently, unfortunately, in my car. This is the best location on the island that I'm currently living in that gave me signal. So hence why I'm here. But I will start sharing in just a moment. There we go. My computer is quite slow. Is that working okay?
Yep, it's working fine.
Perfect. Okay. Hopefully I do not cut off. I have my phone at the highest place that it can get signal. But yeah, thank you all for being here today. I'm going to be presenting an entire presentation, kind of walking you through what it's like to write a research paper. This is based off of my just recent experience writing for ESCOM based off of the data that was collected in 2025. So basically, a bunch of other people were able to collect data, specifically Camilo and Everami, thank you so much. And I was able to go through it for my independent research project in association with Dalhousie. So I just finished that, and Camilo had the wonderful idea of kind of walking the public through how that idea comes to us, like what is the concept that begins it all, how you even like get on the ground running, and then what are the steps afterwards to actually make things tangible. So to start off, as mentioned before, my name is Kaitlyn Quinn. I'm a fourth-year marine biology undergraduate student at Dalhousie, and previously and currently, I work as a volunteer researcher for ESCOM. I also have previous work as a scientific diver and dive master, and currently I'm in New Brunswick as a kayak tour guide, hence the very remote location and very little internet. So, as I said, crossing fingers it won't cut out. But to begin, I wanted to talk about the scientific process. And for today, we'll kind of be going through 5 main steps. The first being gathering previous knowledge. The second is establishing methodology, or kind of a list or steps of how to do something. 3 will be the actual data collection. 4th is the data processing, which I did all last year. And 5thly is the analyzing of the trends. So all of these kind of combine together to actually make up all of the contents of a research paper in almost that order. So you have the previous information that would then become the background of the paper. You have the methodology and data collection for the methods. The data processing would be the results, and the analyzation would be, of course, analysis. And all of these combine together to create something that we call IMRAD, adding the introduction and discussion at the beginning and end there. So IMRAD is kind of the term we always use as the standard paper or most common scientific paper structure. You'll see this a lot, maybe sometimes with the addition of an abstract, which is just like a brief 250-word summary in case you don't want to read through the whole paper. But in most publications, you will see this layout, and it kind of gives people a nice way to flow through things, especially if you're not an expert in the field. So you have always an introduction kind of stating why we're doing that kind of research, the background of previous research. You explain your methods so that people understand if how to recreate your like exact scenario basically. You discuss your results kind of in a more abstract view, and then you dig into things with your analysis and discussion. And this is, you can see any published paper, it'll probably follow this, but depending on the publisher, they sometimes switch them around. So they'll always be labeled though. So now I bring you to my paper. So this is not published yet. This is just submitted for my research project with Dalhousie. I was very lucky to be supervised by Camillo, and I did this basically all of last fall and winter semester because I broke my finger halfway through, unfortunately. It took a long time to write this up. But at the end of the day, I'm super proud of it. It has 20 pages, including the citations and the appendix, and it's called Coastal Plant Dune Species Data Analysis from the Eastern Shore of Nova Scotia. So digging into things a little bit, of course, we have my introduction over here, as you see, and this is based off of the previous knowledge that we gathered. So for this previous knowledge, we originally had some goals specifically regarding the sources that we were looking for. So this was originally, I should say, it was done in about 2025, about winter, and it was done by Christine and a few other volunteers and I, where we got together and Camillo instructed us to gather all the information that we had and could find in libraries, online, just anything related to our topic. And if we could find anything, we would write it up. We'd try to count all of the relevant sources basically for this and combine it into one sheet, which I will show you later. But there's some goals that we had for this in particular, because we couldn't just be adding everything related to plants, obviously. So the first is we tried to look for a lot of trusted sources. So these could be published in trusted scientific journals or acknowledged by other scientists. We refer to something as peer-reviewed when it is a paper or a publication that has been basically given the stamp of approval by other scientists that we trust. So we try to find as many papers that were peer-reviewed. In this case, unfortunately, it was very difficult because there is not a lot of research done specifically on coastal plant dunes in this area. The second thing we want to look for, obviously, is relevant topics. So we didn't want any sort of desert plants, any sort of marshlands. We just wanted dune plants, and we wanted them to be relatively close to Atlantic Canada. So while I did find a bunch of sources for different dunes in like Europe and sometimes even like Maine and south of the border, not exactly relevant. It's a little bit hard to compare, especially from different regions. So we could include them, but it would always be taken with a grain of salt. So we ideally wanted something relatively close. And then lastly, we care a lot about publishing date. So we would like more recent published sources, ideally within the last 10 years. However, we could also include older papers if we wanted to show a comparison of previous knowledge to see like how far things have changed. But overall, we didn't expect much, especially given how limited things were to begin with. And as a result, these are some of the sources that myself and other ESCOM researchers were able to collect in 2025. So the first, which was our big savior, absolutely love this guide. This is by the Nova Scotia Nature Trust, and it's called The Guide to the Atlantic Coastal Plain Flora of Nova Scotia. Beautiful guide, illustrated, has identification of habitats and whether or not they're endangered and whatnot. It was an amazing guide. Unfortunately, it only features any sort of plant in the southern shore. I'm talking southern of Halifax. So that leaves a huge knowledge gap for anything along the eastern shore. So while, you know, it is in the same province, we could kind of assume that there might be some similarities. We couldn't rely on that entirely alone. So taking that with a grain of salt. The next that we found, which was a little bit closer, is the biological inventory of Cape La Have, I believe, island in Lunenburg County. So a little bit closer, kind of similar. We found that a lot of the geography was kind of similar in this area, but it also included a large portion of different types of coastal environments, like rocky ledges. So not all related to dune plants. And another one that I really enjoyed was the Guide to Coastal Zone of Atlantic Canada. So this was a multi-module project produced by the Government of Canada. And it was good. I will say it was a little bit of an older paper. Like I believe this is about 20 years old. So there are like newer publications that we would hope, obviously. But for some of the foundational stuff that I went a little bit into with the analysis, there was some comparisons that could be made there. Now, in regards to more published sources with like scientific articles, we had a few like this one from the Water Resource Research Center. It was like the morphological, atmospheric, and oceanic drivers of cause of temporal saltwater intrusions on a remote sand island. Unfortunately, it was a bit of a smaller island, I will say, than Nova Scotia, so not all relevant in that case, but there was still a lot of overlap when it comes to environmental conditions that I found kind of not, I wouldn't say influencing, but it was interesting to make those connections. Another one is the early successional microhabitats, which allow persistence of endangered plants in coastal sand dunes. So kind of similar environment, kind of interesting. This one, which was actually really close to being within our own area, so like in the eastern shore of Nova Scotia, this one was just north of that, unfortunately, but it also looked at a dune species that we noticed a lot within our study, and that's specifically Rosa rugosa. But this study focused on the fact that it is an invasive species, and it's been invading a lot of Nova Scotia, especially in Cape Breton. So that was something interesting to keep in the back of your mind. And always, the Government of Canada has published multiple sources. There's one specifically that I will always keep in the back of my mind, and it was a Species at Risk Act. I won't post the whole thing. It was hundreds of pages of the document, but they were able to state a few species that could possibly be within not only our wetlands, but our coastal dunes. And it was also a focus on the endangered species, though. So not likely that we would see, but it's good to keep in mind, because if we did see any endangered species, the Government of Canada would definitely like to know that. And it could become some sort of like ecological niche that we could be protecting. But overall, what we learned from all of these sources is that there is little to no research on coastal dune plant species in Nova Scotia along the eastern shore. As I mentioned before, this is kind of not a surprise. When we went into this, Camillo had already warned us ahead of time and got as to why we even started this to begin with, and that was because we could find nothing. So not really surprising, but all of the current published plant ID guides exclusively showed plants, as I mentioned, located in the southern shore, areas in New Brunswick or even the United States, but for some reason always excluded the eastern shore, which was really surprising to me. And then lastly, the studies on dune species mostly focused on invasive species, as I mentioned, the Rosa rugosa, or endangered species, as most of the Species at Risk Act by the Government of Canada. So overall, it was expected. However, it does make our job a little harder, because when we are going into research, we kind of want to know what to expect, especially if we're going to be identifying these species. Unless we rely solely on apps like PlantNet and iNaturalist, we would have no idea what's there, and we would have no idea how to identify it. So our next steps would be creating a list of potential plant species, and this helps us prepare for the data collection, and we can refer to them for identification help in the field itself. So in the early 2025, shortly after we began this research, the other volunteers and I created the first list, which is this species list. This is version 1 of the list, and it is large and overwhelming. So I will walk you through some of the kind of key goals that we had with collecting information. So on the right side, you'll see that we noted the presence on Plant ID apps, specifically on PlantNet and iNaturalist. If it's labeled with an X, that means that we were able to find people submitting pictures of it within the eastern coast. So some of them, like for example, the Brookside alder, the smooth alder, was located on both of the apps. So we can kind of infer, oh, this will probably be present during our time out on the field. So important to note for later. Next, we have the origin. So basically, is this plant native to Canada or specifically Nova Scotia, or does it come from another place? So we have a few different labels for that. Of course, we have native, we have endemic, meaning that it is specifically only found in this location, such as the Nova Scotia false foxglove, love that one. A few other ones that I don't think you see on this part of the list are invasive. That is usually a foreign plant that has no natural predators and will often overtake native and especially endemic plants and pose a risk for not only other plants, but itself, because with no natural predators, they will continue to eat the resources until there's nothing left. So it's something that the government especially wants to keep an eye on. In the case of Rosa rugosa, mentioned earlier, there's a few other ones on this list as well that we can keep an eye on, but luckily, from the whole list, it was mostly native, which I'm very grateful for. Next, as I mentioned earlier, is related to the endangerment status. So we relied on the IUCN and COSAWIC, which are two different organizations that have their own kind of identification for whether or not a species is in danger or like least concern. So we listed those off as well. This part in the middle is more general: location, category, habitats. Basically, is it found in Atlantic Canada? Is it found in Nova Scotia? What category would it be counted underneath? The three main ones that we would refer to are herbaceous, shrubs, and graminoids, or grass-like ones, which are the three main types of plants that could be found based off of our former research. And lastly, it's the habitat that it was previously found in. So CD would represent, of course, coastal dunes. Then we have wetlands, we have salt marshes, and we wanted to see if some of them were exclusive to certain environments or they had a wider range of things. For example, even the grounds elm tree down here, salt marsh, coastal dunes, and also wetlands. So he can spread out a little bit. Then on our left side, we have the scientific and common name. The reason why we have both is the scientific name, one stays the same, no changes, not very commonly at least, versus the common name. There could be a ton of different common names for the same plant, especially depending on regional variation. My best example of this is the mountain lion. Some regions like to call them cougars, and some of them call them, there's a third one I can't remember, but basically all of these different names for the same animal can make it quite confusing. So we prefer to call things by their scientific names so that we have a nice baseline of understanding and we don't get confused. So the little colored list number on the left side was us trying to decide which plants are most relevant based off of all the information that we managed to gather. And I just wanted to show you this part of the list, even though it's in its very rough stages. I think this was the first version, just because I wanted to show you how difficult it is to decide which ones are essential in our case. Because we had a goal at the beginning to kind of have under 70 that we would like to possibly put into an identification guide, which I'm sure you guys have heard lots about. It's definitely in the works now. But with this first list, I think we had hundreds. It was too many plants to remember. Not all of them were super relevant, but because we had no specific guide pre-laid out before us, we had to do everything from scratch. So it was a lot of picking and choosing, all to result with Plant Species List version 3. I do not know if this is the most up-to-date version now. As of when I was doing this research, which was back in the fall of 2025, this was the list that I was given. And I believe we had, I think, up to 70 species. I'll check later. But we had basically all the same information, including like the protection management, the status, the habitat. But for my research in particular, I actually just focused on the names of them, the category, and also I went a little bit into invasive species, but we had another researcher, I believe is Christine, I hope she's watching this, she focused a little more about conservation methods. So it's good to have this information here, even though I didn't use all of it. It's always open to other researchers. So now that we've gathered all our previous knowledge, it's time to establish our methodology. So methodology is a system of ways of doing, teaching, or studying something. And in this case, it's creating basically a list for other researchers to follow. So the research protocols were originally created by Camillo and Aberami following the completion of our species list. And there's a few strategies that influenced our study, basically. I know that specifically Aberami did lots of research into other plant studies in order to kind of get the best methods. So for example, we use transects. I think we call them segments in this, but it's just kind of like a standard way and an easier way to identify groups of plants without just, you know, putting someone in a field and going in circles. You want to have some sort of guidelines for how to do that. So I will try my best to describe that later. But overall, this protocol was written to help guide volunteer researchers to contribute their own survey data. So hopefully you guys. So by following the same guidelines, it helps reduce error or inconsistencies in data collection. And this is essential for obtaining realistic results, because if each of us was doing our own kind of method, it would be all over the place. So here's the beautiful vegetation monitoring protocol from 2025. It has a total of 10 easy steps, and it includes the setup, the identification, and data input, all in extreme detail. I will not go through it with you today, unfortunately, because that's a whole other thing, but I will mention some of the more essential steps in kind of a general showing. So first, we do something called a belt transect, where basically you stick a pin into the ground, you drag across a line, and every couple of meters you will stick a square called a segment, and you space them out equally to kind of give you a good idea of what is along that line. There's other transects that you can do where more of a line where you go along that line, but for such a big area, especially along the coastal dunes, sometimes we find the big segments or quadrats in those squares a little easier to get a better idea in case, you know, you miss a super rare plant just because it's a little left to your line. So I will kind of walk you through it to give you an idea, because I want to be able to give you a more visual, so when you see the data, you're not as overwhelmed. So alongside, let's say this is transect 1 on our beach, we have segments going all the way up to, I think we had an ideal of about 10. I've only written 4 here. But what we would do is then under transect 1, segment 2, I would then stand over it, or in this case, Aberami, because you did a lot of the data collection, thank you, would look inside the square and say what species are inside of the square. So for this dramatization, let's just say it's a beach pea and rugosa rose that were identified in that square. So the next step is to actually go to the ID list. So to the left of the scientific name, you'll notice that there's a bunch of little numbers. And the reason why we do this is because scientific names are very long and a little overwhelming. I cannot pronounce half of these, even on my best of days. So instead, it's a little easier to associate them with a number. So I would check my list. I'll go, oh, beach pea is Lathyrus japonicus, number 11. And then my rugosa rose is number 20. So we would take those 2 numbers and then put them on our little sheet. Now, we also wanted to include the abundance, or the amount within each square. So for that, I should also mention it's more of the sum of total individuals within a given space. So the amount taking up that. There's a few ways that you can do it, but for this protocol, what was created was a list from A to E, and basically you chose the one that best applied for that species. So if I were to look at this little square and I saw that there's one individual, I'd say A. If there was the same, like 50-50, I'd probably say C. And it just depends on which one you thought was most relevant. The other common way to do this would be kind kind of imagining it as a percentage cover. So if I saw a big square and 100% of it was dirt, I would write 100% dirt. If it was 100% beach pea, I would write that. If it was 50-50, I'm sure you get the idea. The only problem with that is that sometimes, especially for non-visual people, it's a little bit hard to imagine, especially when there's a huge break of multiple species. So we tried to create this to be a little more simple, but I will mention in the future that this can sometimes be just as confusing. But for this case, let's say I was looking at my beach pea in Rosa rugosa, and I decided the composition is 11A and 20D. So now, under Transect 1, Segment 2, we have these two numbers and letters. So we know that the number is referring to the species, and then the letter is referring to the abundance. So that kind of gives you an idea of what we're looking like in this very overwhelming Excel graph. I'll walk you through it, no worries. First and foremost, along the top side, you get all of the basic information. So the beach name is Torbay, the monitor name, Aberami, thank you. You get the date, you get the time, and specifically you get the transect number. So Aberami is a hero and did 21 transects. This is transect number 21. Thank you so much. In addition, you'll notice over there that there's all these segments that we mentioned. So each of these represents that big 1 meter by 1 meter square. And then all of these are the species found inside of them. And as we talked about before, for example, segment 7, there's 4 different species under that, and they all have different like identifications. Yeah, there are 4 different species. So wow, that is a lot for data input, and it is a lot to consider. But at this point, I always want to remind people that science is not a closed book. It is not as overwhelming as school and like science and education is, because everything is open book. You're allowed to ask for help. You're allowed to look things up online. You're allowed to look at guides. I find people get really turned off of science because of how overwhelming it can seem. And, you know, there's people who can become experts in this field and memorize every type of graph. However, you do not need to be one of those people in order to conduct science. And if anything, it is always helpful. I have a list of all the identification numbers whenever I'm out in the field. And even if you forget your own monitoring protocol, you will always have a copy there with you. And science is always about interacting with others and having this level of collaboration and resourcefulness. So it's not just about memorization. So yeah, don't be overwhelmed by the numbers. They can always be referred back to. So now that we've established the methodology, now is the hard part, which is collecting the data, which I did not do. So I will summarize what I was told happened. So plant species data collected was following the beach vegetation monitoring protocol created by Camilo and his research assistant Aberami for ESCOM, you guys. So a total of 8 locations were surveyed across the eastern shore of Nova Scotia, and the species identification was assisted by PlantNet and iNaturalist applications, which are always helpful to have on the field. So these 8 locations were chosen because of their coastal dune presence and distribution across the coastline, which covered a wide range of previously undocumented shoreline. So the 8 locations in total, which you will see on my beautiful graph that I made in the corner there, were Port Hilford, Port Shoreham, Peasbrook, Halfway Cove, Seabreeze, Fox Island, Black Duck Cove, and Torbay, which is a wide range and really amazing considering it was just them collecting this data for that summer. So thank you so much. Gave me so much to work with. So a total of 1,080 observations were documented. Some things to note though, at Peasbrook, it was one of the first surveys done. So only 1 transect was done and 13 observations were written down. I heard that, I should say, it always happens. Like you will notice in any sort of methodology that things happen. You know, sometimes your boat breaks, sometimes the weather is not good. If it is field research, things will go wrong. So there will always be, you know, the chance that you miss out on the opportunity of collecting data. It happens to everything. But on some of the best days, for example, the Black Duck Cove, there's 3 transects and 315 observations in total, which is so helpful. That being said, though, because there's such a significant variation of data points, it would be hard to compare the data between these sites, because obviously 315 versus 13, that's a big difference. So it's hard to accurately reflect kind of Peasbrook if it's only represented by 13 observations. So another example of this is how much it would affect the data is that on average, 153 observations per location as a total, all of them, all 7, I should say, locations. That would be the average. But if we included Peasbrook, it would be 136. So docking 20 entire observations just from that average made it quite apparent to us that we should have Peasbrook removed, unfortunately. Love Peasbrook. I hope we can come back to you another year, but for this research, it is better to count it as an outlier in case this impacts our data. So an outlier is considered a data point that lies an abnormal distance from other values in the data set, and this can possibly cause the data to be skewed, or as you saw before, not really reflective of what's actually there. It's not so much that the absence of data is not a reflection of, there not being anything there. It's just that we didn't get to it, and things always happen. So that's important to know. And I will be mentioning Peasbrook again in the future, just because a lot of my graphs, 8 is a much better number, I should say. And it's much easier to have 8 on a board than 7. So he will still be present, but just keep in the back of your mind that it is an outlier. It doesn't reflect what is there at Peasbrook. So that being said, my beautiful graph again, this took me so much time, ignoring Peasbrook, so sorry about that. But now, this is actually where I stepped in again. So starting the data processing, this was basically my entire research project. I started in the fall of 2025 when Aberami and Camilo gave me all of their data and said, go crazy with it. So me and another Dalhousie student, I believe it was Christine, did different research projects. They focused more on conservation status and marking things as, I don't know, endangered, invasive. They did a bunch of cool stuff with their research, and I focused more on the end of creating a baseline with the data that was given to me. So I primarily used RStudio. So RStudio is a wonderful language, basically, for statistical communicating, computing and graphics. But there's tons of other ones out there. I will show you a general amount. It is overwhelming, and they're all very different. However, RStudio was the most common one, at least in my experience, used at Dalhousie in my program. So that was the one that I was most comfortable with. But if you are ever interested in learning a language, it is its own language. It is remarkable what you can do with these things. So unfortunately, it didn't necessarily look like all those cool hacking videos. It was a lot of trial and error and uploading to Excel and removing stuff. And it was a real pain, especially with a broken finger. But basically what you can do with these languages is you can input all the data and say, what does this show me? And you can give them specific mathematical instructions in order to get answers about what's actually there. So this is very overwhelming. I'm so sorry. This is actually my layout for RStudio. I just wanted to jump scare a few of you guys and make me feel better about all of the hours I spent on this. I will not walk you through this, have no fear. I just wanted to show you it because the most important part is first transferring data to RStudio. So usually I wouldn't really reference this. I would have just gone straight into analysis, but I wanted to explain how I got to the analysis in the first place. And the most important part is transferring the Excel data sheets into a form that RStudio can read. And what it can read is something called a CSV file, which is, comparing the two, I'd kind of imagine like the Excel worksheet is a high definition, cool PDF that has a bunch of interactive tools. And a CSV file is like a physical notepad, like with pen. It is the most basic bare bones thing. However, it makes it super easy for RStudio to kind of build it from the ground up in order to make cool analysis. The reason why I'm mentioning this is that this pipeline was one of the biggest struggles that I had, and I came across a huge difficulty, especially with our methodology. So as you mentioned before, I mentioned, the abundance is recorded on a scale that ranges between A to E. And those are obviously alphabetical. Those are not numbers. And unfortunately, we had to completely eliminate these because our studio is not able to understand this arbitrary scale. If it was able to be, of course, converted to a number per se, then it could possibly work. But because we picked this, even though it was with the goal of making it easier for others, we ended up making it more difficult for ourselves. And that's okay. Honestly, a lot of these methodologies and these baselines that we create for ourselves, it is a growing experience. We don't expect everything to work out perfectly. We just kind of have to roll with the punches. So in the future, I'm sure we'll come up with a much easier way to do things that is better for our studio and for the public. But for the moment, I unfortunately had to remove these. But that's okay. In that case, we still have the information of what species were there. So instead, I counted them as individual observations rather than a percentage of each meter. So now we have to choose how we want to analyze. Once we are given our studio and this entire computing language, there's so many things that you can do, and it is kind of overwhelming. So we have to always go back to the basics and think about, what do we want to learn from this data? Do we want to compare locations? Do we want to average things out? Do we want to isolate anything? And how would you do that? So the three main topics that I wanted to focus on were, one, the composition of all the data. So what was the most common plant? What was the least common? What average plants were at each location? Basically the composition of each location. The second is the diversity. I focus specifically on species richness, Simpson and Shannon diversity indices, which I will talk about later. And lastly, I wanted to do a big comparison. So I specifically use a NMDS scaling ordination. I will talk about that later, but I wanted to help visualize comparisons of the species compositions to kind of understand how each location differs. So to begin, for the compositional analysis, I wanted to start off easy. So I just wanted to count the species that were there. So species count is basically the total observation of each plant species at a specific location. And I have this not pretty graph. It's not my best, I will say. This was one of the earlier graphs that I created with RStudio, but I think it gets the message across okay. So this is from the location of Black Duck Cove. It's spelled rock because I spelt it wrong, so sorry. But on the x-axis, which is alongside the bottom, you can see all of the plant species listed in their scientific name. They're not in alphabetical order because I wanted to specifically order them and see which ones had the highest observation counts. So as you can see on the far left, Ambrosia artemisifolia, something like that, is the most common one. And on the y-axis, you can actually see how common it is by the observation count. So I believe it is around 61 to 62 specific individual observations of Ambrosia, which is super cool. But this kind of gives you an idea of just Black Duck Cove. So how about we view it on a bigger scale? So now we want to do species counts for a wide variety of locations. The thing to note is, of course, Black Duck Cove is in the top left, and all of the species names that I've taken out, obviously, so you can actually see this, would be on the x-axis. And these are no longer in alphabetical order, or sorry, they're no longer not in alphabetical order, because I wanted to give everyone an idea of how to visually see the differences between them. So just visually, you can kind of see that Fox Island, Port Shoreham, Port Hilford have somewhat of the same compositions, like some same species are appearing. Of course, poor Peasbrook is in the top right corner, just really fighting away there with those 13 observations. We love that for them. Black Duck Cove, obviously, though, has the most, still 153 observations. No, 315 observations. So obviously more than everyone else. You might be noticing as well, Port Shoreham has about 65 of this. It is unfortunately substrate, so I do remove this at a later period, but in this graph it is included. So substrate is just like dirt. So not super relevant for our case, but I still think this is a cool way to visualize the differences, especially if there's any sort of huge gaps compared to others. But this is obviously just based off of the eye of the beholder. This is based off of me just looking at it. So it would be much better if we had a more, concrete statistical way to tell the differences. So time to get a little more refined. I also did the top 5 species, or at each location, what are the 5 most commonly found plant species. So here's this beautiful graph. We have the addition of a index on the right side, which separates them by color, because now we don't have to worry about a ton of plant species. We only have 12 in this one. And as you can see, there's some color overlaps, especially substrate. Please ignore that pink. But aside from that, some of the more common ones that you'll notice are the Pecal edentula, which is this pretty green. We also have Lathyrus japonicus, which is the beach pea, which we mentioned earlier. And some locations, surprisingly, have some top 5 that aren't even located at others, especially if you notice this Rosa virginia, can't pronounce that. Anyway, that is only found at Peasbrook, which is super surprising because that Rosa Virginia, which is the common name, is actually our native plant here. Like, it is a local rose bush, which is great. Sad that it wasn't located at many of the other locations, but better to see that versus Rosa rugosa, which is the invasive version. But this is super interesting to just see some of the variations. But now let's actually get some more tangible numbers. So I wanted to focus on, at first, species richness, which is the number of species per unit area. And you might be wondering, how on earth do we do that? Because we lost abundance. Well, we technically still have the individual observation counts, as I mentioned before, of each species. And we also have the measurement of area based off of each segment. So if we imagine each segment as a 1 meter by 1 meter, 1 meter squared, we can basically calculate it off of that. So here is the species richness by location. On the left is the percentage of species richness, and on the bottom is each location, which is also colored differently for each of them. No surprise, Black Duck Cove has the highest species richness. Love them for that. And then, interestingly enough, every one of them, except for Peasbrook, don't look at Peasbrook, was relatively similar, which was kind of surprising to me. I thought there'd be a lot more variation. So the fact that they all kind of had the same level per area was quite surprising and hopeful. But we also want to see what kind of diversity that they have amongst them. So I specifically chose 2 main types of diversity indices. And basically, they're both very similar. So one is Shannon and the other one is Simpson. People often get them very confused, but the way to remember them by is basically during the same time in biological history, these 2 people separately wanted to create a diversity index, and they each created their own, named it after themselves. And they do kind of overlap in the sense that they focus on the species richness, which we mentioned earlier, and also species evenness, so how evenly distributed they are. So they use those for their calculations. However, Shannon diversity is better weighted for rare species, while Simpson diversity is better weighted for dominant species. So I will show you the calculations. We do not have to do them. That is the beauty of RStudio and computing languages, who will do all of this for me with a few lined entry. But now we can see this beautiful graph based off of that. So Shannon is in red and Simpson is in blue. The way to kind of refer to this, the index value will be on the left side. They both have different numbers that they base off of. Basically, the higher the Shannon number, usually anywhere in nature, is typically between 0 and 3.5. But all of ours our locations actually had above 2, except for Peasbrook. No surprise. But an indice above 2 is a really good way to say that it's quite actually diverse. There's quite good evenness, and a great way to kind of see this is just a random, I found this on the internet, but these show different community compositions. The first A, you see a lovely array of different rainbow colors. Community B, a little more red, moderately uneven, and then community C is highly uneven. So in the case of community A, if you look down here, super like stable, nice evenness, this would have a really high Shannon indice versus moderately uneven. It would be, this one's about 1.8, it's about the middle. If you have a very low evenness, you would have a very low Shannon diversity number. So on to Simpson though, the max is actually 1. So in this case, if you had a score of 1, that would mean infinite diversity. And if you had 0, it would mean no diversity whatsoever. So the fact that all of them are kind of close to 1 is a pretty good sign. Obviously, are we surprised that Peasbrook is still the lowest of all of them? No. But what is interesting enough is you do see Black Duck Cove, obviously the leader, but there is some variations of diversity levels within some of the locations, which is super cool because we saw beforehand the species richness was kind of dead same all around, but this kind of shows us that there are some more dominant species than others. So now for my favorite, the comparison, I use something called the NMDS, which is the non-metric multidimensional scaling ordination, which is such an overwhelming name. I'm so sorry for that. Basically, I want you to imagine if you had a like beautiful little graph, you'll have a bunch of little points on it, and it doesn't matter what the X is, it doesn't matter what the Y is, it doesn't matter about where they are in space and time. All that matters is the distances in between each of them. So over here we have our lovely ordination, but what you want to notice specifically is each dot is a location, and the distance between them is how much they differ in species composition. So for example, Black Duck Cove is off in its little lonely corner over here, because it is distant from Port Hilford, Torbay, and this whole area, that means that it is not really compositionally the same. The plant species makeup of it is not super similar. However, it is more similar to Port Hilford and Torbay than it is to Fox Island, because that is a farther distance. In comparison, as I labeled with this huge little green square, these 5 locations are quite similar to each other. They're closer. So that means they probably have the same amount of like beach pea ratios. They probably have the same amount of, I'm trying to think, Rosa rugosa. The more similar it is, the closer it will be. And of course, we have Peasbrook in the corner over there. So sorry for that. Interesting, though, that it is close to Port Shoreham, because geographically, I think it is somewhat similar. Yeah, it's hard because we're just going to count that as an outlier, but it is interesting. And of course, Black Duck Cove, unfortunately, because it has so many more individual observations, it is just off the charts for everything else. But what I find most interesting is actually comparing this ordination graph by the actual geographical locations themselves. So I wanted to point out, interestingly enough, Black Duck Cove, Port Hilford, and Torbay are all along, I'd say the southern shore. They're not the southern shore. This is all the eastern shore, but they're more along the same line of the Atlantic Ocean than all the other ones, which are more focused within the Chebucto Bay. Yeah, that's the Chebucto Bay. Yeah. So kind of interesting that they're all within that little triangle. It kind of shows that they maybe are facing the same environmental factors alongside this coast. And then if you look over here, we have 5 of the locations, excluding Peasbrook, Halfway Cove, Seabreeze, Port Shoreham, and Fox Island are all kind of similar. Not really surprising though, because Port Shoreham is kind of distant if you can see over on this right side, but that could be made all the difference by this huge separation in water versus these three are all along the same shoreline and not broken up by as big of bodies of water. So potentially that could have something in it. So you might be asking, what does this all mean, Kaitlyn? This is a lot of information. There's a lot of cool graphs. I really like the ordination, but why is this all occurring? And unfortunately, I don't have the answers, but I can certainly not predict, estimate, I'd say. I can analyze what I see and I'm going to do the best with it. So some of the possible trend explanations, I came up with about 6 potential answers. First would be the geographical location. As we mentioned before, there is some clumping with the ordination that could hint that there is some level of location that makes them a bit more similar. It could be that some plants are easier to like migrate to different areas. Of course, with plant reproduction, some of them have to be physically close in order to spread. Some of them might have seeds that can survive in the water. It changes a lot of how those plants will spread out. So closer geographical locations could have similar compositions because of this. However, I do not have any definitive answers, but it is an idea in the back of my mind. Another could be the environmental conditions. I think this is one of the biggest ones at play, just based off of like personal experience. I'm sure all of you have seen a lot of the beaches, even though, you know, we count them as coastal dunes, they can have such variation. I mean, some of you have already mentioned that it is snowing where you are today, which is insane, but Nova Scotia ultimately is such a big province. And even though on a map these look close together, there is kilometer separation. So they could experience different like weather patterns. They could experience different rain and like coastal erosion, like the water hitting the shore. All of these could impact what plants are able to survive there and could ultimately affect what is making up that area. Another thing that is very common is actually human impacts. Fortunately, I think a few of these beaches were a little more isolated. Sorry, Camilo and Aberrami, for having to walk out into the middle of nowhere. But the lack of pathways, cars, dogs, any sort of like human-related interaction actually makes it a lot more reflective of the natural environment, because a lot of plants cannot survive being trampled, surprisingly. So that could have a huge impact on the composition. Another thing is climate change. We all know that the storms are getting worse, especially here in Nova Scotia. So even some of the beachfronts, especially such a delicate dune environment, would be so impacted by the repeated storms that we've been having the last couple of years. So it is a huge concern, especially for the ones that are more exposed to the ocean and like not in protective little coves, that they might be hit directly by these hurricanes or any sort of coastal erosion with sea level rise. And another one is, as I mentioned before, plants like Rosa rugosa, any sort of invasive species, once they're able to spread and set root in an area, they can outcompete native plants. So that might impact what is there. And lastly, disease and illness. Who's to say? I haven't found anything specific, but I've seen some mention of some plant diseases or even just more invasive species. Like there are certain trees and shrubs that are being taken out by some beetles. And specifically this one little guy, I've been seeing him this year. He's on my mind. So these things could obviously impact either select species or entire ecosystems super, super hard, especially at certain times of the year. But one I wanted to focus on is actually something that I could find other research on that connects to this, which makes me super happy. And that is the presence of eelgrass. So it was mentioned by Camilo, and it was kind of mentioned offhand that some eelgrass was washing up alongside the shore and then somewhat mixing in with the dune environment. And even though it was kind of mentioned in a passing comment, it was interesting that especially at Black Duck Cove, all of the species composition was so different from other locations, and it just so happened to be one of these places that there was a ton of eelgrass. So I found in a few of the Canadian module literatures, especially this one, the module 1, that they have mentioned this before, that the addition of washed up eelgrass could contribute to a lot more what they consider exotic plants, or plants that would not usually be able to live there, are able to thrive because of the excess nutrients. So super cool, could be an explanation, but I'm not too sure, because ultimately, at the end of the day, all of these are very much interacting with one another and impacting one another, and especially for understanding individual factors, it would take a lot of research, and specifically research focused on isolating any of these factors, which unfortunately I do not have the resources currently to do. And this study was just kind of focused on creating that baseline of information. So we don't have any other research to base it off of from previous years. So we can kind of just, make some guesstimations. But my guess is that everything is interacting with one another. So as for my analysis, I'll save you from the many pages that I wrote, and I'll give you kind of a general overview. So of the Eastern Shore Summer Data Collection, they identified 47 plant species in total. Most of it was Hemiphilia breviculata, aka beachgrass. So beachgrass is a grass-like graminoid. It was observed 155 times across all 8 locations, which is really cool because it's considered an essential plant for dune stabilization. So the only location without beachgrass in its top 5 species was Port Hilford, but this could possibly indicate a later successional stage. And this was included in my background, but I just did not have the time to focus on it as much. But basically, the creation of beach coastal dunes, which I'm sure you guys have known from personal experience, especially if you've seen the coastline, they don't form overnight. It takes a long period of slowly encroaching plants building upon the sand and developing an environment that other plants can survive. So the first are considered the primary species. So that's like our grasses. They're able to survive in like sand that has no nutritional value, but by them setting up and stabilizing that sand and creating those like root systems, they allow for secondary species like more shrubs and herbaceous species to then grow upon them. And it kind of, you can see almost like a progressional stage, but the presence of tons of secondary species and less primary species like grasses could indicate what stage of succession that it is in. So the second most common species, though, was Lithyrus japonicus, which is beach pea, which is interestingly a species that can only grow in environments that are previously stabilized by grasses. So even though this grass is quite tolerant to salt spray, this herb is tolerant to salt spray, it can only grow on a place that has already been colonized by grasses. So the beach pea abundance, however, I would like to say that it is a definite sign of a secondary stage of succession. However, the abundance of it varied by location. So some of my thoughts would be that it might be competing with other herbaceous species. It's hard to know though. So overall though, based off of the categories of the most frequent plant species, there was 6 herbaceous, which are usually the intermediate or secondary. There's 3 grass-like graminoids, including our lovely beachgrass, and there's 1 woody shrub, which is a little unsurprising. It is very hard for shrubs to grow, especially in the very flexible, very high-moving, and variable dune environment. So for future studies, as with the main goal of this research, this study can act as a baseline for future studies. And we could expand our research to include repeated testing at the sites that we've already done. We could have new locations alongside the Eastern Shore, and we could record more environmental conditions, like considering whether or not any more recent extreme weather events might have impacted the amount. Even just having like the baseline of what we've seen from previous years, if we then let's say this summer, go to the same beach and we notice a severe lack of certain plants. Oh, we can kind of determine, was there a frost? Was there a hurricane? Was there anything that might have, eliminated that overnight? And that would just be a great way to refer back to information and build upon it for the future. So as I mentioned before, some sources of error, which is always the case with any sort of science. The first is always the potential of human error with identification and data input/entry. Even though we do so much preparation and so much work is being put into an ID guide, at the end of the day, it is all up to the person. So having an ID guide will help to address some of these concerns, and using apps like PlantNet and iNaturalist will help. However, it'll always be a concern. Grasses are hard to identify, so we do what we can. So we also work on creating another abundance scoring system, as I mentioned before. We want to make sure it is still inclusive and easy to understand for beginners and people who are just getting into identification. But ideally, it would be really nice if we could include it and have it able to transfer to RStudio to save me a lot of time. That'd be very helpful. And lastly, I would love if we included GPS coordinates for the beginning and end of every transect. That way we could possibly go back to them each and every year and do the same spot to see if there's any sort of variation over time. So overall, I know that was a lot. Thank you so much for listening so much and for being here. You kind of just listened to the entire process that I went through alongside Camillo and all the other researchers, and everything that went into creating this paper. And it seems really silly, but all 5 of these steps were basically what I did to create my final paper. Wow, that is a long transition. So yeah, following the layout mentioned before, I used all of my previous knowledge for the production. I then created the methodology, I input the methodology based off of what Aberami and Camillo created. I didn't collect the data, but I was able to then do the results and analyze everything. And all of that, everything that I mentioned today is written in this 20-page paper. A lot of people might think it is more complicated than that, but no, really, you just look at the data, you write what you think, you connect it to other sources, and you go, I think I did this wrong, and I think that we should do this, and that was my whole paper. So I hope it was kind of easier to understand, and I hope you don't mind that I showed you RStudio, but I love to hear any questions that you guys might have. Had so much fun writing this paper, and I'm so grateful to Aberami and Camillo for doing all the research so that I can get this done. So yeah, I hope you guys found this interesting and learned a lot.
Yeah, thank you so much, Kaitlyn, for walking us through the process of writing a scientific paper, and also walking us through the really interesting observations that you made regarding diversity and abundance on our beaches on the Eastern Shore. So now's the question period. We're open for questions. So if you guys have any questions, feel free to raise your hand, just as Christina's doing, or just put them in the chat box. I'm just going to start with questions that have already been sent in, specifically from Carolyn. Could animals that eat certain plants affect diversity in different areas?
Oh, absolutely. I especially know for there is a lot of beaches, you'd be surprised, that are so human like interacted with. For example, I'm just thinking off the top of my head, like Rainbow Haven Beach, or even I think there's another one, a few around Peggy's Cove, that have so much human activity and dogs that scare away the wildlife. So I think a lot of the times there is more plants there sometimes, even though there is trampling. But yeah, for more isolated areas where the animals feel more comfortable to go out, I could imagine there's a lot of deer that would go absolutely crazy for those plants. And especially for more of the delicate ones, like the herbs and the grasses, I think would be a lot more at risk. A lot of the shrubs tend to have spikes and like some repellent in that sense, but I can imagine it would make a huge difference. So if we get the funding, I would love to set up a trail cam and maybe we can monitor it long term. Thank you for that question.
Christina, go ahead.
Hi, Kaitlyn. Thank you for your presentation. I really enjoy it. I guess I had some I'm not presentable, but I just for to make sure you're speaking with a with a human being. And I don't see myself. I kind of have a piled up questions. Regarding the first, like the issue of the abundance, the way that you say that it is like not, it wasn't possible to include in the results, and I was wondering if is it not possible to have like make a standardized assumption about the categories used for the abundance in order to include them in the results? Let's say, I don't know, if you had already the category of the dirt, that is, it goes into A, B, C, or D, and you could subtract like assuming a percentage for each category, a percentage of coverage in the quadrant for each category, and then you could subtract the dirt from the rest of it according to the amount of species that they were in the quadrant. So I mean, if is it I don't know, good practice in a statistical analysis to make like a standardized assumption in order to translate this a numerical information into a numerical information for the sake of the program, of the RStudio program?
Oh, thank you so much for your question. I absolutely agree. But yeah, I think what you're describing for the percent coverage is my ideal. I love a percent coverage, especially for collecting information like abundance and comparing to others. And yeah, having it be numerical and like doing a percent. So like even not even writing the percent, but even just writing like, oh, like half of it is this, having designated numbers associated with it would make it easily one-to-one, the transfer of information so much easier. I think what was hard for the one that was done with the abundance, like that scale, is that we couldn't decide upon any numerical equivalent. And that made it so arbitrary. But yeah, in the future, I am highly recommending either we stick to the, percent coverage with the squares, or if we find that's a little too difficult and people are finding it hard for especially such a large area, to determine how much of it is substrate, how much of it is plant, and so on, maybe sticking to even then transects and doing it maybe every 50 centimeters, what plant is underneath that and touching, which would raise its own issues and its own problems, especially for plants that are very unkind for measurement and do not like to be under one spot and tend to move and grow in weird ways. But we'll see what we can do for the future. My heart is on percent coverage because I think that would be the easiest, but we will see whatever is easiest for the main group. But yeah, thank you so much for that. I completely agree.
All right. Camilo, go ahead.
Okay, thank you, Abrami. And first, I want to, congratulate you, Kaitlyn. I think this is a beautiful, amazing, very clear presentation. I think you are a real science communicator. I think it was amazing how you present. Science, it doesn't need to be complicated, as you explained, but at the same time, it's not, it's not a cartoon. You need to have data and you need to explain things that are quite complex concepts. I think you have done an amazing job. I really like and enjoyed your presentation. And one of the things that raised my mind, but I would like to, to confirm with you, you think it could be possible because you have been experienced the situation is, one of the plans that we have now in ESCOM is that we want, to start, the idea that we started in some places in May, but ideally in June, July, to start applying the methodology. So ideally in September, we're going to have data from many other beaches. One of those will be the same beach that we have from last year. Other will be new beaches. Do you think that it will be possible that some student, like you were in this case, can work together with one, the citizen scientist? So just to give you an example, Caroline, she has been always very interested, and she also studied biology, but she will be very interested maybe to collect the data, but maybe she won't have the time, the capacity and everything to do the analysis by herself. So maybe could be a student from biology that could, start working with her, or will be on Anne or Stuart or any of our citizen scientists. Do you think that this could be possible, or what could be the limitations that we could have to start creating this, group of researchers, the academic researcher that will be the student, the biology student, and the on-field researcher that will be our citizen scientist?
Well, I'll first off start by saying, at least with other organizations and other projects that I've seen with citizen scientists, it is absolutely possible. I think some of the limitations would be setting a standard of like assisting for the beginning and like giving that baseline of education, especially for something as difficult as identification. It can take a long time for people to get comfortable. But luckily, with the addition of apps like iNaturalist and PlantNet, it does make it a lot easier, and it could kind of eliminate that uncertainty. Because if they were necessarily just taking down the information on their own and then submitting that data, and we don't see the plants ourselves, we could, have huge sources of error. Like some people could just not be able to tell the difference, or not be referring to the same resources and the same ID guides as us. However, I know with the app that we're working on, and even just in PlantNet currently, contributing the specific pictures from the surveys to groups that we are in, could heavily like eliminate all of that uncertainty. Because if you're taking a picture of the transect and you are saying, 50% beach bee, 50% substrate, and the researcher who is able to actually go in and look at this photo and go, oh, that is not that plant, that is, I don't know, a monstera, that would not be here. They would be able to see that directly and make that correction. And through helping that correction, not only it doesn't make the data input itself more correct, but it also helps the individual learn for next time that, oh, that's actually not a beach pea. It is a long process, I would say, and it would take some level of meeting. It would be ideal to do stuff one-on-one, but I think once individuals, I wouldn't like to say passing tests, because I don't like the education system with testing, but having certain amount of practice and like communication with the researcher before doing the data collection on their own, I think would eliminate a lot of the potential sources of error and a lot of the problems, especially if they're just uploading it and we can refer back to that. And it would cut down on the time extremely, because even like all the time that I saved from you and Aberami collecting all that data, like I got to sit at home and fall and just go through that. So I'm extremely grateful for it. But yeah, it would take a lot of interlapping technology that I know is not always available to us for now, at least. I'm really hopeful for the app that ESCOM is developing, especially after the trial. I have big dreams for that.
I have my old taxonomy books from biology, so I pair it up. But the grasses are very difficult.
No, the grasses are a nightmare for any, like, I see memes on the internet all the time where it's like, I can identify any plant, and then it's grass, and it's no grass. So yeah, taking pictures of it would be super helpful. So even like, I know on PlantNet and iNaturalist, I always post stuff and I wait for experts to reply on it. And I get really sad when it's not the right thing, but you got to learn somehow. But yeah, oh my gosh, if Carolyn, if you could join us for the research, that would be incredible, because ultimately, as Aberami and Camilo know, collecting it is half the problem, especially with such a big province. Love Canada, way too much shoreline and way too much to be to be done. So having everyone feel comfortable and have enough resources to go do it out on their own would be such a time saver and make things more involved.
Just a small comment that I just remember that Caroline, she was in Port Hilford that year when it was she and Abirami who were taking doing the field trip in Port Hilford.
Oh my gosh. Well, I'm sorry I didn't recognize your service, Carolyn. Thank you so much for the data you sent to me. I've appreciated so much of it.
I was the one that identified the difficult to identify plants.
Genuinely, she corrected a couple of my identifications where I was like, oh, it's the same thing, right? No, it's not. Deeply unfortunate, but yeah, absolutely. Anne, you have a question, go ahead.
Hi, Kaitlyn, congratulations. That was awesome. And your enthusiasm just makes everybody want to jump on the bandwagon and get to the beaches. And Abby, thanks for doing all that groundwork, you and Camillo. So one of the questions I had, if we were taking the pictures of the Transact and we couldn't find it, we couldn't identify it with our booklet, for lack of better words, and we didn't have Caroline to kind of go over it. If we took the pictures and just submitted those, would that be accurate enough for it to be used as a sampling, I guess?
It's a good question. No, it totally makes sense. It's hard to say, especially now that I'm thinking back to it. It depends on the size of the segment, because now that I'm thinking about it, a 1 by 1 meter square for something as thin and flimsy as grass would be a complete nightmare. So I'm not sure. Maybe we'll have to like update if we do include pictures in the identification process, it might be kind of an important step to not only take like an above shot, but then individual shots of the plants as well, just to get a better idea. Because I know some rose bushes, they cover everything and you cannot see what's actually underneath. So having someone take a picture of the big transect to get the kind of the greater idea of the percent coverage, and then maybe individual pictures as well of each of them in that segment would be helpful.
Okay, because where we live, often, excuse me, depending on what cell phone you're using. So I have a Bell. There's not very many Bell towers. So I can't go and get my PlantNet or my iNatural to always work. So then it's just a picture. I'm going to take it home. I'm going to try to figure it out. So if there was one that I knew for sure, I don't know this. And if I take a close up and if I identified anything on it, would that be enough?
I'm pretty sure it would, because I know I'm hopefully the app that Camillo and the others are working on will incorporate an offline mode. I'm pretty sure it was mentioned in one of the presentations that you can take the pictures and it will save to the app itself and then be able to be uploaded and identified later. But just to be certain, I'm not super sure if that has been incorporated in the app yet, but I think it can be done. And for the more general, like PlantNet and iNaturalist right now, I think it also does the offline mode, but I know it would be difficult because then you don't know if it's a part of a specific transect. So I think that'd be something important to note for future methods for especially remote areas, because then maybe it would be better to do a transect, just a transect and no individual squares. So you follow a string and you can take pictures along that string and not have to worry, oh, is this within the first section or the next? Because it's all along one string. But that'd be something important for us to have a discussion upon and see, because a lot of these areas are so remote. But yeah, I see your hand raised, Camillo. I'd love to know. Oh, sorry, Carolyn.
Oh, and the stage of growth. So when they're the first two leaves that are emerging, it's hard to identify one plant from another in early growth stages.
Mm-hmm. The only other thing I wanted to ask too was, if the species is endangered, would that be noted on iNatural or PlantsNet? Would that come up, or is there a way to look? Is there a government site to go and look at that has endangered, and then that could be part of the surveillance?
I have to double check. I'm pretty sure if you go, like, when you upload it, I don't think it gives you much information, but I think if you go to the plant like page itself, it will sometimes mention, but I will double check that right now. I have not been on it in a while. Japonicus plant. I'll see, it should include it, but who's to say? And while I search that up, Camilo, do you have
I think
anything to add?
I will start from the last thing that Anne mentioned, is that in our guide, in our identification guide, we mention if the plant is native or if it's non-native, and if it's invasive or not. So this is the way that we have in our guide and our identification guide, that, and it goes to the other comment that I wanted to mention, is that I think the idea to take the picture of the quadrant or the segment is good as a validation information. It's not, I think it's not for doing the quantification or the filter. It's not that you go to the beach and take pictures and that's all. Later you will identify, because it will be so difficult, because plants, plants are not like a garden that you have tomatoes on one side and onions on the other. No, they are all mixed. And as Caroline mentioned, and I also wanted to mention, is that plants, especially here that we have this very short summer, they start growing, very fast and at different paces. So as you can see now, in May, just an example is dandelion. It is growing and now it has flowers. And most of the other plants are just, starting growing. And there are other plants that at the end of the September is when they have the blossom. So it will depend. Each plant is growing at different pace at different time of the season. So it's quite difficult, So it goes to what I think is the most important here, is that I think the only good way to do good identifications is training, is practice. So this is why we are saying this year we want to start. We are not expecting that during this year, the idea that if we go from June to September, it means that we're going to go 4 times, only 4 at the end, is not many. But in these 4 times, maybe the first time that we go there, we won't be able to recognize 80% of the species. So we will need to take picture of all of these species. And when we go back home with internet, go to PlantNet. I always recommend more PlantNet than iNaturalist, because in PlantNet, first, it's based on only plants. Second, you can choose the part of the plant that you are trying to identify. Third, you can add different pictures with different parts of the plant, so it would do a better analysis. And third, that it gives you the different option of plants and the accuracy. So once that you have that, you can go to the guide, because we did with Aberami the first identification, and this is part of what Kaitlyn also explained, that we were based in all this amazing work that was doing by Kaitlyn, Sophia, Christine, that they were, trying to identify all these species. But the idea is that the first time will be quite difficult. Second time, you will identify more species. And, in August, you will say, okay, I can identify half of the species. And maybe in September, you say, I can identify 80%. So it means that you are quite trained. So next year, we're going to start having less error, because you already identify plants. Just to give you a quick sample. Nowadays, I can identify just in one side, beach pea, the Caquilidentula, the sea rocket, and also I can identify the Hocaina piploides, very easily, just seeing one time. But now I'm trained to do that. So my invitation also is that when we go there to our beaches, the idea that we have 3 transects, and because we have this segment meter by meter, at the end of the season, we have been identified, 100 times, the same species. So we will be very well trained to do our job.
Oh, thank you for sharing that, Camilla. That's really good to know. And I just checked PlantNet as well. Unfortunately, it seems that PlantNet does not state whether or not they're endangered. But if you search up the species name and endangered, it should, the most common ones, I believe, was the Kosowick and the IUCN. And they have a bunch of different websites. And if they don't appear on those websites, because those are the specific ones I know in North America, then it's probably not endangered. But all of the ones that are like at least critical or like even not concerned, they will be listed. But there is a lot that are just not assessed, period. So they might not appear there as well. But sadly, yeah, PlantNet does not have that, which is unfortunate. But it's so good to know that on the identification guide that's being built, it will be included, because it is, I think, really helpful to know. And yeah, I'm so sorry if I cut out randomly. My computer has warned me that I'm low battery, and so I will go as long as I can. But yeah, thank you so much for your explanation, Camilo. Yeah, it would be so interesting to see how, I know BHP is like on site for me now, but I still need to get good with my grasses. But yeah, I think it would just be practice, and hopefully yeah, we can have some sort of easier way of like taking the pictures and uploading them. I hope PlantNet can it do it offline too, where you can like save it to the, oh, I think I see you nodding. That's good. But yeah, if that's the case, saving them offline and then referring back to it would be super helpful. But yeah, at the end of the day, any sort of observation is super helpful. And even if you can't identify it, having it be saved and uploaded is super helpful. So yeah, I hope you're able to do some more this summer, and good luck with the grasses for everyone, especially you, Caroline. I'm praying for all of us for the grasses. It's very difficult.
All right, everyone. Well, Kaitlyn, your battery is unfortunately running out, so I want to thank you all and just wrap this up. Thank you all for coming to this webinar today, especially you, Kaitlyn. Thank you so much. It was really interesting to hear your process and all your thoughts on everything related to the beach vegetation. This recording will be posted on our YouTube channel. You just search up ESCOM webinars. You can watch it. Rewatch it as much as you'd like. But other than that, I hope everyone has a good day and a good week, and I'll see you all later. Thanks so much for coming out, folks.
Thank you guys for attending.
Thank you.